Ultra, and that's what he's going to talk about. I was hoping, I remember this paper because it has recordings from a bearded dragon in it, which I find really extremely cool. I hope we get to see those today. So over to you. Hang on, I'll give you a microphone. Right. Yeah. Thanks for coming.
Of course.
Looking forward to hear from you.
All right. Thank you very much, Matthias. Good morning, everybody. My name is Andrew Shelton. Over the last couple of years, I've been contributing to the NeuroPixels Ultra project at the Allen Institute, which has been a joint project between Janelia, the University of Washington, the Allen Institute, and IMEC. And today I'll be presenting on NeuroPixels Ultra, which is the latest iteration in a line of NeuroPixels products to record from single units in the brain. So I think everybody in this room should be very familiar with this paper, but I like to begin here because it's the baseline of what NeuroPixels technology is and why it was necessary as an advent in neuroscience to record from neurons in the brain. Existing extracellular probes before the advent of NeuroPixels were great. They really provided high spatiotemporal properties, but unfortunately, because of site spacing and other aspects of their design, produced low yield in neural recordings. To circumvent this, around the same time, optical calcium imaging was developed to be able to record from many neurons across a given region, but it had high spatial coverage at the cost of low recording speeds, and also can't access aspects of brain activity, such as local field potentials. So NeuroPixels technology offered high spatiotemporal resolution while compensating for that low yield site spacing trade-off, and covers a really large volume in the brain. And just to put some numbers into your head, for a NeuroPixels 1.0 probe, the nearest site spacing is approximately 20 microns on the diagonal. At the vertical, the site spacing is 40 microns, covering a total distance across the vertical shank of the vertical distance of the shank of about 3.5 millimeters. And with the advent of NeuroPixels 2.0, that distance became a little bit smaller, but with better site spacing, better yield, better coverage. So today I'm going to be talking about NeuroPixels Ultra, which is the next generation of these probes. And I really want to emphasize, as in the left side of the screen here, that it really was this massive collaboration amongst a bunch of very impressive people who came together to produce a probe that really tried to compensate for all of the shortcomings of previous generations of NeuroPixels. And I just want to say for the audience online that I'm very gesticulative. And so if I'm pointing at something on the screen and you don't know what that it is, just say so I can use the pointer. So we had three main goals when we were doing this project. One was to characterize the physical qualities of NeuroPixels Ultra in comparison to NeuroPixels 1.0, which is the field standard for systems neuroscience at this point. We wanted to identify whether this improved recording bias for sampling at high density. And we wanted to see if there were any scientific aspects of these probes that they improved, such as cell type specificity, classification, our ability to record from subcellular structures, et cetera. So I'm going to be covering all of these topics today, beginning with motivation for the probe design, physical characteristics of the probe, and work on from there. Also, I also enjoy people asking questions during the talk. I know that we have a section for it at the end, but if you have a question, just feel free to raise your hand and shout it out. So beginning with motivation, why do we need smaller electrodes? And I think, I covered this a little bit at the beginning, but there's really three main reasons why we want to have smaller electrodes encapsulated here. So the first one being this concept of missing units within the brain, missing neurons. And this can happen through a couple of different phenomena. One that was really captured well by Shai Sholem in his 2006 review, coining the term neural dark matter. We have two aspects of the brain that's really difficult to capture in neural recordings. One is that we might be missing neurons because they're just small and they slip between the site the contacts on a given probe because the spacing between them is so large. The other one is low activity. We can't force neurons to fire more unless we're optogenetically activating them. And so we might just be missing a whole bevy of units within the brain because they just don't fire all that much. A second thing that we want to improve with higher density sampling is electrical field sampling. And this is really important for being able to differentiate certain cell types. For example, cortical pyramidal neurons are constructed such that they have electrically isolated compartments, and being able to sample accurately from all of these compartments across the whole dendritic tree will make it easier to identify certain cell types from others. And I think both of these two topics feed into the last topic, which is cell typing accuracy. Our ability to accurately differentiate between different cell types in the brain is dependent on our ability to sample them. And so if we can sample at higher spatial density, we can differentiate between cells in the brain more accurately. And I think you might be able to guess from this point onward, it's going to be kind of a biology-focused talk. I'll talk about physical aspects of the probes themselves, but from then on, I'll be focusing on the scientific aspects mostly. So introducing NeuroPixels Ultra. This is a new generation of NeuroPixels that is based on the previous technology using a NeuroPixels 1.0 head stage, flex cable, and base. The key difference is in this spacing and size of its individual electrodes. So as you can see from this graphic here in a NeuroPixels 1.0 layout, we kind of have this checkerboard design with 12 by 12 microns spaced at different intervals. NeuroPixels 2.0 improves on that by reducing the site spacing, but not the site size. And now with NeuroPixels Ultra, we decrease the site size and the site spacing by an order of magnitude. And so we can fit the same number of electrodes within a much smaller space, increasing the site sampling dramatically. So at scale, this is what a layer 5 cortical pyramidal neuron looks like with respect to a NeuroPixels Ultra probe. And you can tell that we have this unprecedented level of spatial sampling around the soma. And to be a little more explicit about it, NeuroPixels Ultra probes have 384 contacts, 5 by 5 micron electrodes, 1 micron gaps, so a 6 micron pitch between centers of contacts. And newer versions that we're developing now have smaller sites and greater vertical span. And we also provide a switchable version. So this is an electrically switchable version between this 8 by 48 grid layout that you see here to more linearized versions of this probe, such as 4 by 96, 2 by 192, and 1 by 394. So you can kind of get some back some of that vertical span that you lose with the grid layout. And I'll touch more on that later if I have time. So this is a quote from Nick Steinmetz that I found particularly evocative about these probes, is that this device effectively comprises an implantable voltage sensing camera that captures a planar image of a neuron's electrical field. And so I do think with technology like this, we're approaching imaging more than we are electrophysiology, which I find to be very compelling. Okay, so enough of all of that. Let's see some actual data. So here's a 100 millisecond clip from some sorted data or from some raw data I extracted about a year ago. And so on the left here is that 100 milliseconds. The striations that you see in the data is because the probes are laid out in a grid. And so when we unravel them, it looks a little striated like that. And on the right is all of these sorted units from this 100 milliseconds and their spike times. I just want to get you an appreciation of just how beautiful this data really is and how much of an image you're getting of neuronal activity. And so you can see, there's certain units that are bigger and smaller, some that project, have an action potential that looks like it projects linearly versus ones that are more spherically shaped. And I think this is a really great example of the types of data that these probes are capable of capturing. They're excited about it too. and we don't have to play the whole thing, but again, I just wanted to emphasize, when you have effectively a 4-dimensional vector for every unit, the types of data that you're capable of capturing becomes so rich. So in this case, it's X and Y in space through time with voltage. With respect to the physical characteristics of these probes, we found that both in saline and in vivo, NeuroPixels Ultra, despite their smaller site sizes, are actually really comparable in terms of noise to NeuroPixels 1.0. And we found that the increased impedance of these probes did not affect their noise characteristics very much. With respect to photo artifacts from the photoelectric effect, we found that NeuroPixels Ultra have a several order of magnitude lower photo artifact than NeuroPixels 1.0. So this really increases their utility in terms of sorting when you're doing optogenetics using these probes in vivo with NeuroPixels 1.0 and 2.0 probes. Oftentimes what you get are units that are actually just photo artifacts. And so with these probes, we hope that we can significantly reduce the number of units that are classified as photo artifacts. Alessio. We think that the photoelectric effect for NeuroPixels probes is likely due to the CMOS switching technology between different banks of electrodes. And because for the first iteration of these NeuroPixels Ultra probes, we have all of our contacts on one bank, we don't need to use any of the CMOS switches. And so therefore the photoelectric effect is that much smaller. Did I see another hand in back? Cool. All right, so to be able to compare different aspects of these probes to NeuroPixels 1.0 on a unit-by-unit, one-for-one basis, we deploy a technique that we call spatial resampling, and it's where we take a NeuroPixels Ultra-like unit or data collected with NeuroPixels Ultra, and we effectively just interpolate or resample to a lower site density. So in this case, you can see a unit that is taken from a NeuroPixels Ultra recording, and we downsample it to a NeuroPixels 1.0-like recording. And on the right, you can see what this waveform looks like when that resampling occurs. In this case, I feel like this is a very good example of a unit that is relatively pretty small in its spatial evolution of its waveform that actually slips between the electrodes of a NeuroPixels 1.0 probe. And so as a result, the detected waveform by the probe is much smaller in amplitude. We also compare to NeuroPixels 2.0-like configurations, as well as a hypothetical large dense probe, which tests the effect of site size versus site spacing. In this case, the same site sizes as a NeuroPixels 1.0 probe, but the same site spacing as an Ultra probe. And what we see is that for NeuroPixels Ultra, they improve over NeuroPixels 1.0 probes significantly in terms of amplitude, the detected signal to noise ratio of the units recorded themselves, and also have a 3-fold greater unit yield per distance recorded in the brain. So in the case of unit yield, what I want you to pay attention to is this plot on the right, comparing a yield between the different probes, and that middle bar on for each of the probes is the real comparator, showing both stable neurons in the visual cortex that have visual coding properties, therefore implying that those units are real visual cortical neurons. And so I think, as a takeaway, we can say that NeuroPixels Ultra probes have a 10x increased site density compared to previous NeuroPixels Ultras or NeuroPixels probes, so much denser recording sampling. And that the grid layout aspect of NeuroPixels Ultra, it really provides the user with the ability to sample at high density, a given area of the brain, like a cortical layer or CA1 of the hippocampus or the claustrum, if you're me. And so it really gives you this powerful new tool in your electrophysiology toolbox. With respect to the physical characteristics of the probe, again, lower or similar levels of noise, while lower photo artifacts, as well as improved amplitude detection, yield, and signal to noise ratio over previous generations of probes. So next, I'll move on to the types of units that we can detect with these probes, what their waveforms look like, and the possible identities of some of these units. So first, I want to give you a primer about some of these plots that you're going to see a lot of over the next few slides. And when you have this high dimensional data, you want to be able to accurately represent it in a pretty comprehensive low dimensional way. And so we developed these plots to look at, which are essentially a trinity of a spatial, spatiotemporal, and then temporal waveform. In the upper right, in that colorful contour plot, this is the average waveform, essentially smashed out over time. So you're looking at the waveform as it attenuates in space. So in that plot, what you're seeing is just the X and Y of the probe itself. In the plot adjacent to it, that red and blue plot, this is a spatiotemporal plot. So we take the column in the spatial plot that contains the highest amplitude channel, and we spread that out over time of about 3 milliseconds. Actually, I guess maybe about 2 milliseconds. Red colors indicate the initial depolarization of the neuron. Blue colors indicate its repolarization. And then on the bottom is a type of plot that I think a lot of us are very familiar with. This is the 2D temporal waveform for these units. And so here we have displayed a regular spiking unit and a narrow spiking unit detected by NeuroPixels Ultra. Any questions about these plots? I know, like I said, you're going to see a lot of them, so I want to make sure everybody's comfortable with that. Okay. But again, I think there's a lot to be gained out of visualizing waveforms like this, but it's also important to realize that, again, these are evolving in time and there's some multi-dimensional aspect of them. And so we wanted to develop features that accurately capture the evolution of these waveforms in time and space. So there's very classic metrics that we can use, like the duration, the peak to trough ratio, the amplitude to describe, certain aspects of the unit's waveform. But we also wanted to develop metrics like spatial footprint, which capture how much the unit spreads in space. And so we defined this as the radius around which the mean waveform amplitude decreases to 30 microvolts. And we chose 30 microvolts not arbitrarily, but because there's a 10 microvolt noise floor for these recordings. That's just the baseline noise for NeuroPixels Ultra probes. So we wanted to choose a threshold that was significantly above that noise. And so what this looks like is when we calculate it for a given parvalbumin positive unit here, is we swing a line around the maximum amplitude channel and ask at what distance does it fall below 30 microvolts on average. And so for this unit, in this case, it'd be about 80 microvolts or 80 micrometers. And we look at a distribution of almost 10,000 units collected at the Allen Institute from the visual cortex. We see this very interesting bimodal distribution of unit footprint sizes split at about 20 micrometers. So when we surveyed units from across the brain, we found that this bimodal distribution could be found in almost every area, most notably in visual cortex, but also in parts of the basal ganglia, the thalamus, but that this relationship was really pronounced in places like the dentate gyrus and the corpus callosum that had this large abundance of very small units. And so in these Trinity plots that you see for each area, you can see a representative large and a representative small unit. And what you can see is that the waveform for all these small units is highly stereotyped. So we asked, what could these units be? When we looked at the distribution of these units with respect to their footprint size and duration, we found that the vast majority of these small footprint units, these very small, teeny tiny units, had very fast waveforms. And that when we broke out our distribution by duration, we found that regular spiking units did not represent these small footprint units by and large, but that these units formed this bimodal distribution primarily within the fast spiking unit population. And so to kind of parse this out a little bit, in extracellular electrophysiology, if you have a narrow spiking waveform, typically people think that you're an inhibitory neuron. And so we wanted to determine whether that was actually the case. And so in 3 different transgenic lines, we optotagged inhibitory units of 3 different flavors. And here's just some statistics from these small fast spiking units, making up, roughly half of all fast spiking units detected in our recordings. And so we did this in both in all 3 of parvalbumin, somatostatin, and VIP inhibitory transgenic lines, covering the main cell classes within the inhibitory group of cells. And what we found is that although each of these classes had a small component of fast spiking small units, they didn't overrepresent that population of units more than the others. And that when we tested these small fast spiking units for connections to other units, we found that they had a bidirectional relationship in the sense that some of these small fast spiking units could either excite or inhibit their postsynaptic partners. And this was done using a cross-correlogram analysis. And so to really test what these units are, now suspecting that they might not be inhibitory units, one of my collaborators, or my co-author on this paper, is Ziwen Yi at the University of Washington, applied Mucimol over the visual cortex during NeuroPixels Ultra recordings. And Mucimol as a GABA agonist would inhibit the cell bodies of any recorded units within that area. And so what you can see from this spike raster here, these are all units on the y-axis, time on the horizontal axis. That is that at Mucimol application, the vast majority of all the units recorded become silenced, with the exception of a select few. And those select few are all of our small fast spiking units. Given that this is the case, our suspicion now is that these small fast spiking units are locally recorded axons whose activity is not dependent on cell body or somatic firing within a given area that we're looking at. And so what this tells us is that NeuroPixels Ultra have such a high density of electrodes within a given area that we're looking at that we substantially increase the number of axons that we can record from a given area, which is incredibly impressive. So not only are these probes extremely good at recovering unit activity with high yield from small areas, such as single cortical layers, cortical nuclei, thalamic nuclei, but that if you're very interested in studying fibers of passage, such as the corpus callosum or elsewhere, these really are the probes for you. Okay, and so finally, given that I have a few minutes left, I'll talk about some fun use cases, one of which is covered in the first iteration of our paper, cell typing, and the second of which, backpropagation, is covered in this now next iteration of our paper. Cell typing, just as a primer, is really important for being able to describe neural circuits because the brain is composed of many different cell types that each uniquely contribute to how the brain performs a given computation. And so within the visual cortex alone, which is covered here in this Gowan's 2021 paper from Cell, you can see that there's this really broad variety of different excitatory and inhibitory types, each with their own waveform and firing pattern, and presumably their own computation to neural computation. So being able to describe what a neuron's type is from its extracellular waveform is kind of like the holy grail of extracellular electrophysiology from that standpoint. And so being able to accurately say what neurons are which within a given extracellular recording is incredibly important for being able to say what that area that you're recording from is actually doing. And so we asked 2 main questions of this schema. With what accuracy can we predict cell type from NeuroPixels Ultra recordings compared to NeuroPixels 1.0 recordings? Right, and I guess what is the difference between the accuracy of those 2 probe types? So again, we're using the spatial resampling technique that I mentioned earlier. And so again, we return to our inhibitory transgenic lines for PV, SST, and VIP to assess the accuracy, forming a true positive label matrix for our recordings. So here are like grand average waveforms from each one of these clusters. And in this scheme, we were using random forest classification over both the temporal waveform as well as extracted features, including footprint for each unit type. And so we have PV, somatostatin, VIP, regular spiking, large fast spiking, and small fast spiking units. And this large fast spiking category is composed of all of the fast spiking units that were collected from data sets where we didn't know what those particular units might have been, say in like the VIP data set or something like that. The results of this showed us that through random forest classification, we could accurately cell type using NeuroPixel's ultra probes to the tune of about an average of 70%, which is, you know, good, not the best, but definitely very good. And that we could predict cell type from these recordings significantly better than we could with NeuroPixels 1.0 like data. And that when we included footprint as the single metric of a single spatial multi-channel metric, we again significantly improved performance, showing that adding any information about how waveforms develop and evolve in space is incredibly important for being able to describe the type of neurons that we're actually recording from. And that when we iteratively removed features from the model, say amplitude or duration, we found that spatial footprint was as important as duration in being able to describe the cell type of, or to be able to predict the cell type of any given unit that we recorded. Any questions about this before I go on? I know I'm pushing time. Cool. All right, so finally, I'll talk about detection of subcellular electrical events using NeuroPixels Ultra, namely backpropagation. People have studied backpropagation both from a single cell in vitro electrophysiological perspective, as well as an extracellular electrophysiological perspective. And backpropagation is seen as this very important neural biophysical phenomenon that is important for things like synaptic plasticity, and it has been shown to occur predominantly in neurons like layer 5 pyramidal neurons. So to understand this phenomenon specifically, we expressed channel rhodopsin in layer 5 pyramidal neurons of the cortex and recorded from visual cortex again. Here you can see some very stereotyped waveforms from 2 of these optotagged units in this transgenic line, and you see that they are incredibly similar. Indicating that 1, we're both recording from a specific cell type, and 2, we're seeing the phenomenon that we actually want to see, which is this big red swoosh as it goes up the probe, indicating a traveling wave of electrical activity heading up the apical dendrite. To be able to do this across the whole cortical column and not just the limited set of sites that we get out of the standard NeuroPixels Ultra configuration, we use our ultra high density switchable probes. And these are, again, these probes that allow us to switch into more linear configurations from 8 by 48 to 1 by 384, recovering a lot of span and allowing us to record from that apical dendrite as the backpropagating action potential travels up the dendrite. And so what we found is that we could both detect these events and also detect them at higher signal to noise ratio than in previous versions of these probes, which is a really powerful tool for being able to study subcellular electrical events on a single neuron basis. So this gives NeuroPixels technology an even greater window into the types of computations that single neurons are capable of performing. And so, you know, from here, you could take this to how backpropagation might be able to enhance or might be able to affect neural computation through spike timing changes, et cetera. So finally, just to wrap up, NeuroPixels Ultra trade out in their initial iteration span for improvements in signal to noise ratio, amplitude, sorting accuracy, yield, et cetera. And that we have new versions of these probes that have the same site density, but allow you to recover a lot of that span through linearization, just really improving the utility of these probes. And I think really the takeaway message is if you have your own little area of the brain that you're really interested in getting a very high density, very high yield recording from, this is the technology for you. Finally, I want to thank all of my co-authors and collaborators on this project. as well as Alessio, who provided a lot of help in being able to sort these recordings. Truly, all the work that I've shown here is maybe 10% mine. So I could not have done it without these people, especially Ziwen, Julian Bassard, Jordan Shaker, really very impressive scientists. So thank you for the time. And I suppose, question?
Yeah. So the way this is in get rid of these things.
Yeah.
You say that there's some reliable way of excluding these spikes.
Sure. So what we found with these axonal spikes is that they actually have really variable waveforms depending on the type of axon that you're recording from. A recording from a node of Ranvier might look very different than an arborization of axons as they enter into the cortex. But a key feature that really unites them is that they have a fast spiking waveform that is very small in its spatial extent. And when I looked within the visual coding and visual behavior data sets produced by the Allen Institute, fully 10% of all the units recorded with just NeuroPixels 1.0 probes were axonal in their waveforms. And so if you wanted to remove that contamination from your recordings, my suggestion would be to look at the number of channels that waveform is seen on and just develop some, either bespoke threshold or stick to the, the 20 micron threshold that we found here in our data set and just remove those units that fall below it. Because, if you're thinking physically, an individual neuron, like a pyramidal neuron, will have a large cell body that is at least 30 microns large. And so the waveform that it sets up can propagate for hundreds of microns. And so if you remove that small subset of units that have very small waveforms, I think you'd be well set up for excluding those from your data set.
I have two questions. First is the burning button. When are they going to be built?
So we have new probes, the switchable versions dropping this month. And those will be available at a price of between $1 and $1.5K. So about the price of a current NeuroPixels 1.0 probe. We do. Adam Huntman has done that, and he is in the process of sharing that data. Our next steps through review will be to record from dentate gyrus granule cells and determine the size of the waveforms that those units set up. So it's in the works. Any other questions? All right, thank you again for your time, guys. Appreciate it.
Right, thank you very much. So let's switch over.